Welcome to this visual tour of how Auxin sculpts root systems beneath the soil. The title slide sets the stage for a deep dive into the signals and transport mechanisms that build roots, and why understanding this master plan matters for plant growth and resilience. Root system architecture, or RSA, is the plant's interface with soil, defining anchorage, nutrient uptake, and environmental sensing. RSA depends on root length, branching degree, and growth angle, and its plasticity lets plants adapt to stress, so decoding molecular controls of RSA can help breed crops that use water and nutrients far more efficiently. Auxin acts like a molecular glue that converts local concentration into gene activity. At low auxin levels, AUX/IAA proteins repress ARF transcription factors and keep genes off, while high auxin promotes AUX/IAA degradation through TIR1/AFB, freeing ARFs to turn on developmental programs and change cell behavior. The root tip hosts a stable auxin maximum that patterns the apical meristem. Polar auxin transport via pin carriers creates a gradient that defines the quiescent center and stem cell niche, helping position master regulators such as WOX5 and PLT to maintain stem cell identity and drive primary root growth. The transition from cell division to differentiation is set by a precise auxin-cytokinin balance. Auxin promotes division by degrading repressors like Shy2/IAA3, while cytokinin stabilizes them to promote differentiation, and the resulting transition zone position determines meristem size and sustained root elongation. Lateral roots form through a four-stage sequence orchestrated by auxin. Periodic priming marks future sites, an auxin maximum initiates asymmetric founder divisions, a structured primordium forms with organized cell layers, and finally the new root emerges by breaking through the overlying tissues to connect with the soil. Long before a lateral root appears, an oscillating auxin signal sets a root clock that pre-patterns branch sites. Reporters like DR5 pulse roughly every 6 hours to create spaced pre-branch patches in the pericycle, a process relying on core components such as TIR1 and ARF7 and local auxin conversions in the root cap. At a defined pre-branch site, a localized auxin maximum triggers founder cell specification and the first asymmetric divisions. The SLR/IAA14 ARF7/19 module activates LBD16 to specify founders, and a biophysical feedback from pericycle-induced endodermal turgor loss creates the mechanical space needed for those divisions to proceed. The developing primordium engineers its own escape route by becoming a local auxin source and pumping auxin into overlying endodermal and cortical cells via AUX1 and LAX3. The resulting high auxin activates wall remodeling enzymes like expansins to separate cells and allow the new root to emerge through the tissue layers. The gravitropic setpoint angle, or GSA, shapes the overall root architecture by setting the oblique angles at which lateral roots grow relative to gravity. GSA is actively maintained by asymmetric auxin distribution at the tip, and it determines whether roots forage shallowly for phosphate or grow deeper to access water. Lazy family proteins control GSA by directing the polarity of auxin efflux carriers in gravity-sensing columella cells. In wild-type PIN3 localization produces a steep setpoint, while altered lazy activity shifts pin polarity, redistributes auxin across the tip, and steers root growth toward a shallower angle in lazy mutants. Final root architecture emerges from a dynamic dialogue between a genetic blueprint and environmental cues. Availability of phosphate, nitrate, and water reshapes RSA through modulated auxin biosynthesis, transport, and signaling, enabling plants to forage resources effectively by remodeling their root systems in response to conditions. Plants deploy distinct auxin-mediated strategies to cope with scarce resources. Under low phosphate, they favor deeper foraging with longer lateral roots and steeper angles via enhanced TIR1 sensitivity. Low nitrate often boosts lateral branching near the topsoil through localized nitrogen signals, and water scarcity promotes deeper, hydropatterned roots with ARF7. Mediated regulation to seek moisture. Auxin's versatility makes it the central coordinator of root development, linking primary growth, branching, growth angle, and environmental plasticity. A unified signaling module of TIR1/AFB, AUX/IAA, ARF, and dynamic polar transport yields robust but flexible developmental outcomes that let roots adapt while maintaining core functions. Understanding auxin's control of root biology is essential for building the next generation of climate-resilient crops, because improved root systems can capture water and nutrients more efficiently, reduce fertilizer dependence, and enable breeding strategies that prioritize sustainable yield and resource conservation under changing environmental pressures.